Thank you for joining us today. My name is Jason Chibuk. I am the Vice President of Medical Affairs at Baylor Genetics, and we welcome you today to our webinar entitled From Symptoms to Sequencing, Diagnostic Insights from 3 Pediatric Genome Sequencing Cases. Before I introduce our speaker, I wanted to note that we're going to hold questions and answers until the end of the 25-minute presentation, but you're certainly able to submit your questions along the way at any time by clicking the Q&A box at the top of your screen. So without further ado, I'm pleased to announce our today's speaker, Lisa Salz. Lisa is a board-certified genetic counselor currently serving as a senior medical science liaison at Baylor Genetics. Lisa's career spans 30 years in genetics, and her primary area of clinical focus is rare pediatric disease and genomics. We're looking forward to your presentation, Lisa. Please take it away.
Thank you, Jason. In today's webinar, we'll address 3 objectives. We'll compare traditional genetic testing with genome sequencing, examine 3 patient cases that were resolved with Baylor Genetics genome sequencing testing, and demonstrate the clinical utility of genome sequencing for pediatric patients. Before we describe our 3 patient stories for you, let's review what might be considered conventional genetic testing paradigm, which begins with ordering targeted genetic testing and then proceeding to increasingly more comprehensive testing when the previous test is not informative. Consider an infant who presents in the NICU with treatment-resistant seizures, a cardiac malformation, and dysmorphic facial features. The clinical team may first order a targeted test, such as FISH for 22q11 deletion syndrome. If negative, then they may proceed to a multigene panel or chromosomal microarray. Multigene panels and CMA have a diagnostic rate of approximately 5 to 24%, depending on patient phenotype. Exome sequencing, which has a diagnostic yield of 25 to 40%, depending on the setting and the patient's phenotype, is the next most comprehensive test that might follow. But if this is also non-diagnostic, then what is next? Is there a reason to pursue additional testing? Is there additional diagnostic yield to be achieved? We hope that the patient cases today will demonstrate that the answer to this is yes. To further compare and contrast commonly ordered genetic tests, targeted NGS panels focus on a predetermined list of genes related to a suspected condition and are most effective at identifying single nucleotide variants, indels, and some selected copy number variants. Chromosomal microarray, or CMA, is optimized to detect copy number variants across the genome, including deletions and duplications that are below the level of resolution of traditional karyotyping. Chromosomal microarray can also identify absence of heterozygosity, but it cannot detect single nucleotide variants or indels, which are below the resolution of the assay. Genome sequencing, more comprehensive than CMA or targeted panels, covers approximately 2 to 3% of the human genome and is a more comprehensive test than the NGS panels for detecting single nucleotide variants because it does not depend on a predetermined or condition-specific list of genes to interrogate. Exome sequencing covers the protein-coding regions of the human genome, but does not include intronic or regulatory regions. It also does not include coverage of the mitochondrial genome. While exome sequencing can detect copy number variants, its sensitivity is generally lower than chromosomal microarray or genome sequencing. Copy number variant detection on exome sequencing is limited, particularly for smaller CNVs or those outside of the targeted exonic regions of the platform. In the final column here, genome sequencing is the most comprehensive genetic test available because it covers 98% of the nuclear genome, including exonic, intronic, and regulatory regions, as well as coverage of the mitochondrial genome. This allows for robust detection of copy number variants and detection of uniparental disomy when trios are available. Genome sequencing also includes coverage of selected genes associated with short tandem repeat disorders. Overall, Genome sequencing provides up to a 20% increase in diagnostic yield over other, more targeted genetic tests, including exome sequencing, depending on patient phenotype, age, and the quality of previous testing and sequencing. Several professional societies, including ACMG, most recently the American Academy of Pediatrics, NSGC, and the American Epilepsy Society, endorse the use of genome sequencing as a first-line test for clinical indications, including multiple congenital anomalies, intellectual disability, developmental delay, and seizures. The clinical utility of rapid genome sequencing is increasingly recognized as a valuable diagnostic tool in the acute care pediatric setting because it avoids the need for sequential genetic testing, allowing clinicians to initiate targeted medical management within a few days rather than a few weeks or months. Let's review several patient cases for which genome sequencing provided a molecular diagnosis that other genetic tests would likely have missed. Our first patient we will review today is a 4-year-old male who first presented to genetics clinic at 18 months of age with an unusually shaped head, mild dysmorphic facial features, and failure to thrive. At that time, imaging revealed a large arachnoid cyst in his right cerebral hemisphere, which required a shunt. He was next seen at 4 years of age in genetics, at which time his genetics evaluation revealed ADHD, autistic behaviors, and delayed speech and language development. The patient's family history was unremarkable, and he had had no previous genetic testing. The genetics clinic ordered duo genome sequencing at this visit. Genome sequencing revealed an intronic variant of uncertain significance in the CHD5 gene. CHD5 plays a role in facilitating chromatin remodeling and gene transcription and is associated with autosomal dominant Parenti-Mignon neurodevelopmental syndrome. The initial study on this condition by Parenti et al. In 2021 identified 16 individuals with CHD variants. Common features include speech delay, behavioral issues, epilepsy, dysmorphic facial features, motor delay, impaired intellectual development, hypotonia, and several of the patients had craniosynostosis. It is noted that disease expression of this condition is highly variable within families. The reported variant has not been described in ClinVar and is considered rare with a very low frequency in NOMAD. This intronic variant was predicted to cause acceptor gain and alter splicing. However, the functional impact of this change remained uncertain at this time. We know that variants of uncertain significance present a challenge to clinicians in making informed management and treatment decisions, so additional information would be needed and helpful to upgrade or downgrade this variant. Because this variant was predicted to have an impact on splicing, it qualified for clinically validated targeted RNA sequencing called RNAseq at our laboratory. So RNA was extracted from an additionally submitted sample, amplified for the region, including the CHD variant, and sequenced by next-generation sequencing. The result of RNAseq for this patient is shown in this sashimi plot, which is a visualization tool to display alternative splicing events. RNAseq demonstrated that this patient's CHD variant causes abnormal splicing, resulting in an out-of-frame partial intron retention in the mature RNA. You can see this in the case and the case repeat images as compared to the RNAseq control image. The RNAseq assay showed that this variant results in abnormal splicing and introduces an out-of-frame deletion and a protein with a premature stop codon. Thus, targeted RNA sequencing provided sufficient functional evidence to upgrade the variant to likely pathogenic, and an addendum report was issued to the ordering provider. The reclassification of the variant from VUS to likely pathogenic established a molecular diagnosis and enabled the clinical care team to focus its care, including monitoring for the possible development of seizures. RNAseq provided a diagnostic answer that would not have been identified with traditional testing approaches. As was illustrated for this patient, RNA sequencing is a powerful tool to assess the functional impact of DNA variants that are predicted to affect splicing or gene expression. One very large study looked at nearly 690,000 individuals across 9 clinical specialties. 5.4% of patients in this cohort had a splicing-related VUS. Of those, 1.7% were shown to be reclassifiable using RNA sequencing. That may seem small in terms of overall diagnostic yield, but when you consider the size of the rare disease patient population and the clinical significance of reclassification for just one patient and family, this can be a very meaningful number. For our second patient, she is a newborn female who we'll describe today. She presented in the NICU with hypotonia, respiratory insufficiency, and hypoventilation. Intrauterine growth retardation was noted prenatally. These nonspecific features presented a broad differential diagnosis, including spinal muscular atrophy, metabolic and mitochondrial myopathies, Prader-Willi syndrome, and muscular dystrophies. In the genetics consult note, it was stated that given the critical nature of the patient's presentation, rapid broad testing is indicated. Thus, trio genome sequencing was ordered. Genome sequencing revealed maternal heterodisomy for chromosome 15. Maternal uniparental disomy occurs when an individual receives 2 copies of chromosome 15 from mom rather than a copy from each parent. The Prader-Willi critical region, 15q11 through q13, on chromosome 15 is inactive on the maternal chromosome. Because mom's contribution is silenced, dad's contribution is necessary for the genes to be expressed in this region. This finding on genome sequencing is consistent with Prader-Willi syndrome. The estimated prevalence of Prader-Willi syndrome is 1 in 10,000 to 1 in 30,000. The clinical features associated with the condition evolve over time. In the newborn period, these infants have significant hypotonia and poor feeding. As they grow, features like developmental delay and excessive food-seeking behavior is observed. Individuals with Prader-Willi syndrome also have a distinct behavioral phenotype, short stature, and mild intellectual disability. As you can see in this image, Prader-Willi can be caused by several molecular mechanisms. 70% of individuals with Prader-Willi have a deletion in that critical region, 15q11 through q13. Approximately 28% have maternal uniparental disomy for chromosome 15, and about 2% have an imprinting defect in this region. There are no phenotypic features of Prader-Willi syndrome correlate exclusively with any of the 1 molecular mechanisms that result in Prader-Willi, but there are some statistical differences in the frequency or severity of certain features between patients who have the 15q deletion and those who have UPD15. Historically, diagnosing Prader-Willi syndrome has required either targeted testing or a combination of multiple tests, each designed to detect a specific mechanism. FISH, fluorescent in situ hybridization, was used to identify deletions, but could not detect uniparental disomy or imprinting abnormalities. Chromosomal microarray is highly sensitive for detecting the classic 15q11 through q13 deletion, and when using a SNP-based platform, chromosomal microarray can also detect regions of absence of heterozygosity, which may suggest uniparental disomy. However, CMA cannot determine the parent of origin, so maternal UPD, the second most common cause of Prader-Willi syndrome, cannot be confirmed with chromosomal microarray alone. Additionally, imprinting defects cannot be identified by microarray. So historically, methylation analysis can detect all 3 major mechanisms, deletion, maternal uniparental disomy, and imprinting defects, but it does not provide information about the molecular mechanism causing Prader-Willi syndrome. Genome sequencing, on the other hand, offers a comprehensive genome-wide view in a single assay. It can reliably detect large deletions, such as 15q11 through q13, and can infer uniparental disomy when trio data is available through regions of homozygosity. However, whole genome sequencing does not assess methylation status, so follow-up testing may be needed to identify the associated 2% of cases caused by an imprinting defect. Most families who have a child with Prader-Willi syndrome have a very low recurrence risk for this condition. However, certain etiologies have a recurrence risk as high as 50% and very rarely 100%. But reliable recurrence risk assessment requires identification of the genetic mechanism of Prader-Willi syndrome in the proband, and in some cases, additional testing of parents. The importance of making a molecular diagnosis for Prader-Willi syndrome is that several emerging and investigational therapies are being developed to address the complex manifestations of the condition, particularly the hyperphagia, the overeating, food-seeking behavior, and the behavioral symptoms. Growth hormone therapy remains the standard of care, improving body composition and physical development. Oxytocin analogs are under investigation to enhance social behaviors and to regulate appetite. VICAT XR, which has shown promise in reducing hyperphagia by modulating hypothalamic pathways, was approved by the FDA in 2025. Finally, there are early-stage studies involving epigenetic therapies aimed at reactivating silenced maternal genes on chromosome 15. These efforts reflect a growing pipeline of targeted treatments that may transform care for individuals with Prader-Willi syndrome. In summary for this case, when ordered for acutely ill patients with a large differential diagnosis and for a condition that has several genetic etiologies, rapid genome sequencing provides a molecular diagnosis that allows for initiation of targeted treatment, identifies the molecular mechanism resulting in the condition, and provides recurrence risk information for a family. For our third and final patient today, we'll introduce a 4-year-old female who presented to clinic with microcephaly, hypotonia, and global developmental delay. She also had failure to thrive and required G-tube feeding. Based on her clinical presentation and concern for an underlying genetic disorder, sequential genetic testing was ordered. Prior to ultimately receiving a diagnosis, she'd had a total of at least 6 genetic tests that were completed, including chromosomal microarray, methylation studies, gene panels, exome sequencing, mitochondrial DNA testing, and biochemical profiling, all of which were non-informative. And in fact, she had a 7th test, which was genome sequencing at Baylor Genetics in 2023, which was also non-diagnostic. However, her clinical team remained suspicious of an underlying genetic disorder, so they requested reanalysis of her genome sequencing data in 2024. Reanalysis confirmed a de novo insertion in the RNU42 gene, a non-coding RNA gene. Non-coding RNAs contribute to a wide variety of cellular processes, including the maintenance of germline genome integrity, the regulation of gene expression, RNA splicing, and translation. Dysregulation of non-coding RNAs is implicated in both cancer and neurological diseases. RNU42 encodes U4 small nuclear RNA, which is a key component of the spliceosome, and it plays a critical role in RNA splicing during brain development. The variant shown in this patient's genome sequencing report is an insertion at nucleotide position 64, which is the common de novo insertion associated with what is now recognized as Renew syndrome. The variant is classified as pathogenic and is predicted to disrupt a critical pseudognat structure, which impacts RNA splicing. Renew syndrome was first identified in 2024, and additional patient cohorts have been published on by several groups now. Renew syndrome is increasingly recognized as an emerging prominent cause for neurodevelopmental disorders, accounting for up to 0.4% of individuals with neurodevelopmental delay. The clinical features of this newly described neurodevelopmental disorder include intellectual disability, developmental delay, seizures, and feeding difficulties. There's now a Renew Syndrome Foundation that has been created for this condition, and they have been vocal about the importance of broad, comprehensive genomic testing in order to ensure individuals receive appropriate and timely diagnoses. This patient story highlights the importance of non-coding RNA genes and the power of genome sequencing to detect variants that would not be detected by other genetic tests. It also emphasizes the importance of genome sequencing reanalysis following non-diagnostic comprehensive testing. Reanalysis of genome sequencing data can provide important diagnostic insights. A general recommendation for clinicians is to wait at least a year to request reanalysis because this allows for the publication of new gene disease associations, as well as the updating of clinical databases and bioinformatics platforms. It could be considered at less than a year, however, with the emergence of a new phenotype for a patient. Studies have shown that the diagnostic yield from genome sequencing reanalysis can range from 2 to 11%. So with that today, with our 3 patient stories, we hope to leave you with several key takeaways. Namely, genome sequencing is indicated when a patient has a broad differential or there are many different genetic causes for a suspected condition. Genome sequencing provides comprehensive coverage and broader detection of variants in a single methodology, with a higher overall diagnostic yield than other genetic tests. Genome sequencing provides more timely diagnoses than sequential genetic testing, which enhances its clinical utility. Targeted RNA sequencing for qualified variants increases the diagnostic potential for genome sequencing. Several professional societies support genome sequencing as a first-tier test for indications, including congenital anomalies, epilepsy, intellectual disability, and developmental delay. And finally, as knowledge advances and new gene disease associations are discovered, reanalysis of genome sequencing data can yield additional diagnoses without the need for additional testing. And to end, today I'd like to just provide a brief review of Baylor Genetics Whole Genome Sequencing offering. It provides genome-wide detection of single nucleotide variants and copy number variants, and the reporting of clinically significant exonic, intronic, and non-coding variants. This testing includes 29 genes associated with short tandem repeat disorders, complete coverage of the mitochondrial genome, the ability to detect uniparental disomy when trios are sequenced, and an opportunity to reflex to targeted RNA sequencing for selected variants. Thank you all for your time today.
Amazing. Thank you so much, Lisa, for your presentation. I see all the applause coming in. I also see lots of familiar names on there. So hello, San Diego and the rest of the crew. The Q&A, again, is up in the top of your screen. There is a little button there. You can type your questions in. I've received a couple of questions through direct message along the way. So I'm going to start with those as the rest of you populate any questions that you have. We have about 5 minutes left for Q&A. So the first one, Lisa, is in Baylor's experience, what is the reclassification rate for RNAseq, Ben?
Okay. Thus far, the reclassification rate has been similar to the publication that I had cited earlier on the very large cohort. We currently have a manuscript under review, and about 4% of all genome sequencing cases will qualify for RNA sequencing. They have an eligible variant. About half of these, 1.7%, have been reclassified, and most of these are upgrades from variants of uncertain significance to likely pathogenic, and occasionally likely pathogenic to pathogenic. And I thought I would also touch, maybe, Jason, to follow on that question, a little bit about what types of variants qualify for RNA sequencing at our laboratory. And the basic qualifications would be, is the variant in a gene that closely overlaps with the patient's phenotype or clinical condition? Is that variant, was it detected on exome or genome sequencing at Baylor? And has there been any other RNA-seq testing performed? That would be an ineligible variant then? And then finally, do the in silico prediction tools predict that it will affect splicing or gene expression? So we use Splice AI and some other algorithms to help detect that. And currently, these are listed on our reports when qualified variants qualify.
Excellent. Very thorough response. Thank you. I think RNAseq is poised to become a very interesting add-on, sort of the next layer of omics, and it's great that that it's available and showing good utility. The next question that I've got come in directly here is a friend who orders exome sequencing through Baylor Genetics. With the additional variant types that genome can find, what do we think the increase in diagnostic yield is of genome over exome? Sort of what's that delta?
Yeah, I think there are a lot of publications that have addressed this. It will depend on the indication for testing, what previous testing has already been done and non-informative, and then whether or not we're able to sequence comparators with that testing. Is it proband only or trio? But in a recent review of our internal data, which has not yet been published, we found that about 10% of individuals who were diagnosed with received a molecular diagnosis with neurodevelopmental indications for testing. About 10% of those received diagnoses that would not have been detected on exome sequencing. And the reason for that is that several of these patients had, or many had, variants, pathogenic variants in deep intronic regions. It was due to mitochondrial variants, pathogenic mitochondrial variants, or expansions in our short tandem repeat genes that resulted in a diagnosis that would have been missed by exome.
Excellent. Thank you so much. So we've got time maybe just for one more, but a question about mitochondrial genome coverage with WGS, which is sort of an added element. What's the coverage like? What sorts of things can you detect? Maybe talk a little bit more about mitochondrial genome coverage.
Mhm. So on our whole genome sequencing platform, we do capture variants within the mitochondrial DNA across the mtDNA genome. And with that, this does come from our genome sequencing data. So it's all in one platform that we're able to assess this. We have a level of detection of heteroplasmy for single nucleotide variants down to 5%, and for copy number variants in the mitochondrial DNA down to 10%.
Excellent. And we have one more that I think is great for the maybe non-geneticists in the crowd. It's a wonderful question. Quote, I know that testing parents helps with interpreting genome results, but can you describe in detail how that happens? I think it's intuitive to some of us, but others might wonder why go through the hassle of getting mom and dad samples as well as the proband.
Mhm. Yeah, no, that's an excellent question. And the importance of having biological parents as comparators for that genome and being able to sequence concurrently allows us very quickly to assess the inheritance of variants that might ultimately be reported for that patient. And what I mean by that is we're able to quickly assess whether or not the variant is de novo, which may add to the classification of pathogenicity, or is it recessive? If we simply have two variants in a gene that causes a recessive condition, we're not sure if that's in cis or in trans. And when we have parents available, we're able readily to assess that, is this in trans? Is this therefore disease causing these variants?
Perfect explanation. Thank you so much, Lisa. All right. Well, unfortunately, that's all the time that we have, friends. So thank you, Lisa, for being such an amazing speaker. And thank you, Lisa, for the presentation. Thank you, audience, for participating. If you have additional questions, there's an email address here at the bottom of the screen, insights@baylorgenetics.com. We'll also send out a link to the recording in the coming days so that if you want to share it with colleagues or review it again, you're certainly welcome to. Thanks again for your time and have a wonderful day, everyone. Thank you.